others, the loops had now become ordered and were in contact with what had been the original hydrophobic polypeptide binding surface that had now moved through space to make contacts with the GRO-ES mobile loops. And so this just summarizes basically what happens. So huge rigid body movement of the apical domains. They elevate 60 degrees and they rotate 90 degrees clockwise. So that basically, if this was the original hydrophobic binding surface, here pointing into the cavity, the movement essentially left the hydrophobic surface pointing out of the board at us and replaced it with a new surface facing the central cavity. And so the effect of GRO-ES binding was essentially to enlarge the cavity. So this was a 45 angstrom diameter of an unliganded ring. And now with GRO-ES bound, it's roughly 60 angstroms in diameter and roughly 60 angstroms in height. But the key is that GRO-ES has now encapsulated this chamber. And so the polypeptide is basically folding in isolation. There's simply nothing it can aggregate with because it's in isolation. Moreover, the chamber walls have changed from this hydrophobic binding surface of an open ring to what's shown here in blue as all electrostatic residues, which we believe constitute a no-stick surface that allow the polypeptide to go about its business of folding without a lot of interference with the walls. Yes, there are translational and rotational motions that must bang into the wall. We've observed that with time-resolved anisotropy experiments. But my guess is that the idea is a no-stick surface so that the polypeptide follows its way down the energy landscape. So fairly recently, I'll just make one little additional comment here about dome formation. In some EM experiments carried out at Scripps with the NRAM collective, we've been able to essentially add ATP to unliganded GRO-EL and watch what happens to it in the absence of hydrolysis and in the absence of GRO-ES. And we see several states that suggest the trajectory. So the trajectory would go as follows. Here's unliganded GRO-EL. It binds ATP, and when it does so, both the apical domain and the intermediate domain together as a unit sort of crunch down on the nucleotide. And there's a slight elevation, for example, of these helices that have the binding surface called helices H and I. They're slightly elevated. There's then a second movement around a hinge that's at the top of the intermediate domain that moves these helices up a little bit more. So now H and I are moved up a little bit more, and that produces this state that we call RS open that we believe is the GRO-ES docking state. So now GRO-ES really comes on, and now you get this huge further change that I showed you a few slides ago where the apical domains elevate 60 degrees and make a 90-degree clockwise twist to produce the final dome chamber. So what's the advantage of this? Well, so this first step where the two domains move down as a rigid body doesn't really allow GRO-ES docking yet, but starts to move things in that direction. The further movement of the apical domains now puts the hydrophobic binding surface in a place where if you look at the GRO-ES mobile loops and try to just directly drop them vertically down, they could now dock to the apical domains, to that hydrophobic surface. And that then initiates this gigantic elevation and 90-degree clockwise twist. So you can see that the apical surface has come around by 90 degrees and produced the dome folding chamber. So polypeptide during this step is released into the cavity to commence folding. Okay, so this is the domed folding chamber. So you can see that it's loaded with electrostatics, blue for basics and red for acidics, and very few hydrophobics are left. We've mutated those and there's no effect on polypeptide chain folding. So the question is this charged hydrophilic cavity actively assisting folding or behaving passively? So there are two ways to look at GRO-EL reactions. One is under non-permissive conditions where there's an absolute requirement for GRO-EL, GRO-ES chamber. So for example, those early Rubisco experiments I was describing to you where Lorimer couldn't see any native material produced at all unless the machine was there are non-permissive conditions. So folding free in solution under those conditions produces no native product and there's quantitative aggregation. But then there's permissive conditions, remarkably, where the substrate protein can fold to the native state either inside the GRO-EL-GRO-ES cavity or free in solution. So I want to note that permissive conditions involve typically lower substrate protein concentrations, for example, 10th micromolar instead of the normal 1 micromolar that the field often uses, or lower temperature, for example, 15 degrees instead of 25. Note that both of these conditions would disfavor aggregation. Okay, finally, and importantly here, because this is where contention has arisen, there are some substrates that can fold up to 10 times faster in the chaperonin system under permissive conditions than free in solution. Is this due to active assistance by GRO-EL-GRO-ES? So I want to show you a number of experiments that make me think that there's no active assistance by GRO-EL-GRO-ES, and that what we're looking at simply is that proteins, even under permissive conditions, do a lousy job of folding free in solution, that they tend to aggregate. So here's an experiment with Rubisco. Folds 5 to 10-fold faster at the chaperonin than free in solution. But look at this. These are two different studies that I won't cite, but they both required BSA in the solution reaction in order to get any recovery of Rubisco and substantial amounts of BSA. By contrast, the chaperonin reaction doesn't care whether it has BSA or not. It goes ahead and folds 5 to 10-fold faster. So Adrian and Petrie in the group said, well, okay, I'm going to do the experiment without BSA and see what happens. And here's what happens. You basically get no recovery. So we think that BSA is probably behaving in a fashion that forestalls aggregation to some degree. Not sure exactly how it does that, but it seems to be crucial. In fact, he could look at this reaction with dynamic light scattering, and even in the presence of BSA, unfolded Rubisco, the minute it comes out of denaturant, produces a strong light scattering signal. By contrast, BSA, or native Rubisco with BSA, does not produce that kind of signal. Okay, a second substrate that can be folded under permissive conditions. The chaperonin-mediated refolding of DMMBP, a double mutant form of maltose binding protein that's become a popular substrate recently, is 6 to 8-fold faster at the chaperonin than free in solution. So here's either a GRO-EL-GRO-ES reaction or an SR1 GRO-ES reaction, and here's a spontaneous reaction. Quite a slow refolding over roughly 80 to 90 minutes here. So Navneet Tyagi, who's here at this meeting, and Adrian have both carried out experiments looking at this particular protein. So Adrian initially carried out experiments on inclusion body purified material, and Navneet has now carried out experiments with what is a little bit cleaner material that is purified from E. coli using an amylose matrix affinity method. So he's purifying the material in the native state, basically. And both of them observe what is a strong concentration dependence of the refolding of DMMBP free in solution. So here there's about a 3 or 4-fold difference in the rate of folding. So as you go to lower and lower concentrations, this protein folds better and better. And so I don't know what else could really describe that other than aggregation. That is classic behavior for aggregation. Here's the behavior of wild type MBP. It has no such concentration dependence, and it folds a whole lot faster than this particular double mutant. Moreover, chloride-free medium was discovered by Adrian and confirmed by Navneet to relieve the concentration dependence that's observed over here. And now the protein, the scale here is a little different, folds about 5-fold faster than even at the lowest concentration here, and is no longer concentration dependent. In Adrian's experiments, what he observed under those conditions was that there was no more aggregation observable by light scattering that he could see with his inclusion body purified material. And Navneet is now doing the same kinds of experiments to see if there's light scattering or what goes on in this particular reaction as evidence of oligomerization. But even more interestingly, the rate of refolding by GRO-EL-GRO-ES in the presence of chloride is basically identical rate to this, what we believe is an aggregation-free chloride-deprived solution over here. So we think what's going on is that GrowEL doesn't have the ability to particularly accelerate folding. It's rather that in free and solution, folding is decelerated by these competing kinetic steps that are reversible. You're making low-order oligomers, for example, that compete with the pathway of producing the native state. There's more work to be done to really prove this, but I'd like to mention an experiment that's been done elsewhere that I think is very powerful. And so there's a prediction from these experiments that at infinite dilution and free solution, the rate of folding in solution will become equal to the rate of folding inside GrowEL, GrowES, if we're right about all of this. So Ben Schuler and his group in Zurich have carried out an experiment with roadmeats using single molecule fret, reported last year, and observed exactly that. The slow folding rate limiting domain of roadmeats could be observed to fold at the same rate free in solution as inside the chaperonin. So to us, this argues for at least this substrate, at least, that there's no acceleration mediated by GrowEL. The protein folds at basically the same rate inside and outside. That's not to say that the walls don't have some minor influence on how domains organize, but the machine doesn't have an intrinsic ability to accelerate the rate of folding. Okay, so I just want to make a few more comments concerning the ATP reaction cycle, and then I'll finish off.